Professor Kell, your talk today has an intriguing title, Don't Do Pathway Analysis Without TRANSFAC. Why such a strong statement?
Well, of course, you can technically do pathway analysis without TRANSFAC, but you risk missing crucial upstream mechanisms. Most tools and databases focus on known protein-protein interactions and downstream effects. TRANSFAC fills the upstream gap, connecting transcription factors to their binding sites and revealing causal regulators. Without that, you often get an idea about the effects, but not the causes.
As someone who frequently uses KEGG, Reactome, and Panther for enrichment analysis, I'm curious. What exactly are we missing with those tools?
They're useful, but they tend to highlight processes like metabolism or cytoskeleton remodeling, effects, not causes. For example, we analyzed data of treatment of fibroblasts with TGF-β, a known inducer of fibrosis. Standard tools missed the TGF-β pathway entirely. Why? Because they don't include transcription factor gene connections. That layer, what turns genes on or off, is usually absent in such type of analysis.
Interesting. So how does TRANSFAC help uncover the upstream regulators?
We start by identifying differentially expressed genes, then scan their promoters using TRANSFAC's positional weight matrices to predict transcription factor binding sites. More importantly, we search for combinations of these sites, composite modules, because transcription factors rarely act alone. These combinations tell us which factors are likely driving the observed gene expression.
But once you have those transcription factors, how do you trace things back even further? Say, to master regulators or drug targets?
That's where TRANSFAC integrates with TransPath, a massive signal transduction network database. We trace upstream from the TFs to identify master regulators. We prioritize those that activate multiple relevant TFs and are part of feedback loops. These master regulators often represent disease-driving nodes. If available, we also integrate proteomics to adjust weights. Active proteins get more importance.
And how do you deal with the complexity of signaling rewiring in disease?
Great question. In pathologies like cancer or fibrosis, the usual signaling pathways get rewired. New feedback loops form and transcriptional control shifts. TRANSFAC and TransPath help detect that rewiring. For instance, even 48 hours after TGF-β treatment, when downstream noise is high, we were still able to recover the original signal using this method.
How accessible is this for researchers? Do we need to be programmers?
Not at all. We built Genome Enhancer, a web-based tool that lets you run this entire upstream analysis without coding. You just upload RNA-seq or epigenomics or proteomics data or all of them together, define your groups, and the system does the rest. It identifies transcription factors, searches for upstream master regulators, and even suggests drug targets using our human PSD database.
And in your fibrosis study, did Genome Enhancer successfully identify TGF-β?
Yes. Despite all the secondary changes that had occurred after 48 hours, the system correctly traced the signal back to the TGF-β receptor complex and mapped its cascade to the observed gene expression changes. This wouldn't have been possible without transcription factor prediction.
It's truly impressive. So, final question. For someone studying gene regulation in disease, what's your one-sentence advice?
Don't just look at what changed, ask why it changed. And to find the answer, start with transcription factors. That's where TRANSFAC gives you an edge.
Thank you, Professor Kell. I think many researchers, including myself, will rethink how they approach pathway analysis after this. I am going to apply Genome Enhancer right away.
Thank you, Dara. Looking forward to seeing your results.